Hi, I am Kaylee Beer. Like you just said, I'm a student at the University of St. Thomas, and I will be discussing the link between dissolved organic matter, or DOM, its composition, and the degradation of selected environmental contaminants. This project was done in collaboration with the University of Wisconsin-Madison, and our funding comes from the National Science Foundation. Our overall goal for this project is to fill the knowledge gap between DOM composition and its photochemical reactivity. Some brief background. Selected contaminants in waters can degrade via direct or indirect photolysis. Our project focuses on the indirect aspect, of which DOM is an important character. DOM is a photosensitizer, which when reacting with photons from the sun produces photochemically produced reactive intermediates, or PPRI, such as triplet excited state DOM, singlet oxygen, and hydroxyl radicals. And these PPRI degrade contaminants at varying rates. And so if you're interested in that, my colleague Alyssa will be talking about that later on. So to look at this relationship, we have collected samples from 45 sites in Minnesota and Wisconsin that fall into 4 different site type categories, and those are agricultural, rural, urban, and wastewater treatment plants, which we actually collected those from all regions except for the Northern Highlands. Okay, moving on to our results, this top graph, briefly to tell you how we got this, we spike our water samples with one of the selected contaminants and then photolyze it for a predetermined amount of time and run those samples through our HPLC for analysis. And so from this graph, which is specific to atorvastatin, we can see that there's a large range in observed rate constants going from about 0.07 for our direct samples to almost 1 for the wastewater treatment plant samples in the St. Louis River region. We can also see that sample site types have a similar rate as well. So Mankato and Yahara have somewhat similars, being those the agricultural. However, we do see that in the Northern Highlands, there seems to be a large range in observed rate constants, and this is also reflected in our carbamazepine data as well. However, when we looked at our sulfadiazine data, we didn't see the similar pattern. We found that the Mankato and Yahara sites had some of the higher observed rate constants, while the St. Louis River sites actually had some of our lowest observed rate constants. Moving on to our carbon normalized rates across the board for all 3 contaminants, we found that the St. Louis River has the lowest observed rate constants, indicating that the DOM and PPRI there likely are less efficient at degrading contaminants than sites like Mankato, which have much higher observed rate constants. Lastly, we wanted to look at the relationship between the concentration of dissolved organic carbon, which we use as a surrogate for DOM, and the rate of degradation. Looking at atorvastatin, we can see that there is a pretty clear relationship. That is, the concentration of DOC increases, the rate also increases. However,
About 1 minute left, sorry.
To level off. Looking at carbamazepine, we see a little bit different relationship. It seems that is more site type specific, having different slopes for different site types, which are those different colors. And then lastly, looking at sulfadiazine, we are unclear on this relationship. We just need a little bit more data here to make a firm conclusion. So overall, the relationship between DOM and contaminant transformation seems to be variable by sample site type. Thank you.